Eugene Myers, Dr. Eugene Myers, you come from what background?
I'm a traditionally trained computer scientist.
A computer scientist, and you're attending today in the University of San Diego, talking about gene therapy and DNA and a variety of gene splicing, a whole bunch of things. How did you get involved in all this?
Well, back in 1985, it was becoming apparent that computation was going to be a critical part of molecular biology, and the reason for that is basically the scale and the volumes of the data sets that are involved. Genomes involve billions of base pairs of characters. There are tens of thousands of genes that produce hundreds of thousands of proteins that interact in millions of ways.
Okay, everybody's faded out now by the time you've mentioned all those things that you had to do. So let's just say it was a very complex task.
Yes.
And your idea was, or not your idea, but I guess it was the world's demand that we break down the human DNA. Is that basically what we're doing here, or what you were doing?
Well, I don't think it was actually the world's demand. I mean, it was basically scientific curiosity that was driving all of us. We felt that if we could sequence the genome, we would get access to everything that was in there. In other words, everything that was encoded, and that we would begin to really understand how cells work, how we work at a molecular level.
Now, there's rumors there. How many are there? 300 million genes, or we don't know any of this. So you're going to have to help us out here.
We believe that in the human genome there are about 30 to 40,000 genes.
30 to 40,000 genes. Okay, and what's a gene? What does it do?
A gene is a region of the genome that is involved in encoding for proteins. And the proteins are molecules that perform functions inside of your cells.
And so everybody's genes are different or similar, or?
Everybody's genes are pretty much the same. We're about 0.1% different from each other. And those 0.1% are very different. I mean, small changes in genes can cause diseases. They can change your tendencies to develop particular conditions. They determine how long you live, what kinds of problems, what your health is. So those very small differences are actually quite important. But basically, we're all the same. We're more the same than we are different.
What are you going to be talking about today at this conference?
I'm going to talk about how we can more efficiently sequence the genomes of a variety of organisms, including people. And the issue there is that if we can sequence, if we can obtain the individual sequence or genetic fingerprint of individuals, the code of individuals, then we'll be in a position to predict and to understand, to basically correlate the people's code with their health conditions. And by being able to do that, we'll then be able to understand the correlation between what's encoded in your genome and what your health outlook is like. And this will be extremely important.
Have you done this with plants and animals as well? Are you doing gene sequencing for plants and animals as well?
We're sequencing the world.
Are you? So now let me ask you a question. I have a plant at home that's not too healthy. Could you do the gene testing on that plant and find out what was wrong with it and why it won't grow as fast as it should?
Do you water it regularly?
Don't ask me those are tough questions.
Let me jump in, Ron.
Yeah.
Gene, you said I'm going to quote, we now have the Rosetta Stone, but don't know what it says. Can you explain what you meant by that?
Well, we actually have the sequence. So we have the individual letters. We have the text, but we don't actually understand how to read that text. We don't understand how to interpret it. We don't know what it says. So it would be like, me getting a text in Hebrew, which I do not speak, right? And if you gave that to me, I wouldn't understand what it was saying. I could say that I have a particular document in hand, that I have a particular message, but I don't know what the message is. And so it's the case that we're partially able to interpret what's in there, but we're not really getting at all of it, and we really need to be able to understand what's in there.
What's the corrective action? What do you think is going to happen so that you can understand it?
Well, it's not that anybody's done anything wrong. It's simply just science is advancing. It's a complicated.
Well, we hear that science is advancing, but we don't. When most people don't understand what means science is advancing, they're looking for solutions to their cancer, their AIDS, their coronary thrombosis problems, or their heart disease, or whatever. So they're kind of confused on where this this energy is going and what it's going to accomplish at the end of the day. Is it going to be able to help people come up with cures for those things?
Yes, it is. It most definitely.
And how does it do that?
As we get further and further into it, we're going to understand exactly what the mechanisms are at the molecular level. And the more that we understand about that, then the more we're going to be able to intervene either with drugs or with gene therapies to basically correct those conditions.
Good word, gene therapies. Can you tell us what a gene therapy is? What is gene therapy? What does that mean?
It basically means reintroducing a correct copy of a gene that you might be missing.
All right, let's say you have either a bad one, let's say you have a bad gene like my plant at home, or else you've got, let's say, a one that's missing. How do you put that back into somebody's body that didn't have it before, or else it's the one that's there is broken?
Therein lies the problem. We don't really know how to do that effectively. I mean, there are ideas around, putting it into, stem cells that go into bone marrow, and there are a number of ideas that are being approached, but that really is the.
So learning how so that your Rosetta Stone story was kind of, I guess, very truthful when you said, "Gee, I don't know how we're going to be able to understand it first, and then how to apply it to the human condition."
Yeah, I mean, realistically, so I mean, all kinds of promises are made. I mean, I'd say that realistically, we're really. It's clear that we stand on the precipice of being able to really understand and improve our health situation. I think it's going to be something that's going to unfold over the next 50 years. Okay, I mean, realistically. I mean,
well, what's your
part? I don't want people to think that this is going to happen tomorrow.
Right. Well,
it's not going to happen tomorrow.
Let's ask this though: what progress have you made over what period of time? What have you done since? When did you start this?
Well, we started
85?
sequencing the. When did I start? What did I start?
Yeah, when did you start sequencing?
I've been a scientist for 25 years.
No, I meant for just the genome is what I meant. Yeah.
Well, we started thinking about sequencing the genome, I think, as early as 1985.
Right.
And the project got very serious in 1990, and then it accelerated. I do think that, so things are accelerating. I mean, everybody understands that the technology is accelerating. And so it's a little bit hard to make a prediction about when, particular breakthroughs might be made. But I really think that it is, the trajectory is clear. And, I mean, people should get overly anxious about, or anticipatory about when we're gonna make these breakthroughs. But it's clear that we are.
Well, what I was getting at is that you started sequencing this project, and then have you seen positive results since the day you started? In other words, by sequencing, were you able then to find a cure for something, or apply something that fixed something?
It's not what I specifically pursue, so unfortunately I can't give you the anecdote.
Well, were you helpful in supplying that to others,
I think
perhaps?
that by having the human genome, by having the Rosetta Stone, and having decoded what we do know about it, okay, and we know quite a bit about what's in there. We just don't know everything. But given that we know about it, we have basically improved greatly people's ability to understand the genetic basis of diseases.
Where do you supply your information to? When you do your research at a university level, do you give it to drug companies, to the government? Where does this information go?
Well, the human genome is available in the public domain. So
it's on the web. You can just pick up, go there,
to the NCBI site and grab it. Yeah.
And so
if you really want to grab it.
So it's totally public domain. Is that right?
It is.
And then so what types of people would access that information for use?
Scientists, primarily. It's rather boring to look at a 3 billion sequence of ACs, Gs, and Ts. It won't tell you much.
But you said 3 billion, and then when we came to genes, you said 30,000.
Well, you can look at the annotation of all of those genes if you'd like to. That information is also there.
So it's 30,000 genes, but it's 33 billion.
3 billion letters in the human genome.
There are 3 billion letters, and inside of there are encoded, we think, about 30,000 genes. So we've basically annotated or noted within that genomic sequence where the genes that we do know about are encoded. I mean, that information is provisional in the sense that some of those gene models aren't correct, completely correct, but correct in outline. Others of them are completely correct.
Let me ask you some supposition. I hate to ask scientists a supposition, but since this is your level of expertise, I feel fairly comfortable doing it. When Dr. Crick and Dr. Watson founded this concept, or developed the concept in 1953, we had a computer called UNIVAC, the Sperry UNIVAC computer. And now, with computer expertise as it is, if Dr. Crick and Dr. Watson were to have found that process today as opposed to when they had the Sperry UNIVAC, how much quicker would they have been able to find all this?
I don't think that the Sperry UNIVAC or the computation would have made a difference in understanding what they did was they discovered the structure, the molecular structure of DNA.
But they were, did the computer help you?
If they had modern crystallographic techniques,
yes,
with modern computers, it probably would have taken them about 2 months.
2 months.
And it could have been done by basically anybody who was competent in X-ray crystallography.
And back then it took how long?
Well, I don't know how long it actually took.
50 years. They didn't have the
But they were
technology.
working hard. I mean, there were all kinds of issues. There were issues of how to actually isolate the DNA. I mean, what was DNA? Figuring out that there was this thing called DNA, actually making crystals of it, getting a good X-ray picture of it. I mean, there were a lot of things that went into the discovery of the structure of DNA.
So, Dr. Eugene Myers, what is your calling at this moment in this whole process? What do you have yet to accomplish in this human genome sequencing that you've been involved with? What are you planning?
Well, I'm interested in taking the genomic sequence, actually understanding everything that's encoded therein, and then understanding how basically it's unfolding within the cell. So I'm very interested in development and basically whole models of cellular function. I mean, I think we're on the precipice in the next 20 to 30 years of being able to build a complete model of all of the agents that are working in the cell, all the molecular agents that are in the cell, and how they're interacting.
Dr. Eugene Myers, thank you for joining us on The Cutting Edge.
Sure, you bet.
Dr. Bruce Alperts, and you're the president of the National Academy of Sciences in Washington, D.C. Is that correct, sir?
That's right.
Well, tell us what we're doing here, celebrating the 50th anniversary of the double helix in San Diego, California.
Well, this is honoring, in particular, Francis Crick.
And who is Francis Crick?
Francis Crick was the partner with James Watson who in 1953 discovered the structure of DNA double helix, which immediately broke a mystery, which was how do we get our characteristics inherited from parents to children.
So he's sort of the modern-day Sherlock Holmes with his real Dr. Watson.
That's right, that's right. And that made so many other things possible. So all the people you see in the audience today, almost all of them, their work is only possible because of the discovery that they made 50 years ago.
Well, let me ask you this. Modern day. Let me ask you this, Dr. Alperts. You've been involved in this process with DNA for how many years?
I first got involved in 1958 as an undergraduate at Harvard doing research, and that's how I heard about DNA.
Incredible. Well, in today's society, people don't understand about 4 different words, and if you'll just, because of your background and the fact that you're really so involved for so many years, could you help us out quickly? You say the word cloning to most people. What does that really mean?
Most people, cloning seems to mean making an identical animal to the one that you clone from. That is, what we call, in the case of the sheep dolly, reproductive cloning, or in the case of the people who want to make a human clone, human reproductive cloning. A big problem is that there are many other ways in which that word is used, and they've been totally confused in much of the public debate.
Do they make plants? Can they clone a
You could
plant?
clone plants, you could clone bacteria, you could clone yeast, you could clone DNA, and so Cloning is a procedure widely used in science, and it's been an important technique for many of the breakthroughs that we've had in the last 50 years.
So it's a scientific sort of Xerox machine that just cranks out people the same, or cranks out anything, animal.
We don't want to crank out people the same. The scientific community has gone strongly on record. The National Academy of Sciences had a major report saying we would like a ban on human reproductive cloning. But we need also to discriminate that from all other kinds of cloning. All those other kinds are enormously important for the advancement of science. And what's happened in the public debate is that whether on purpose or not, these terms have gotten all mixed up, and so people want to get rid of all cloning, or way too much cloning, kind of cloning that actually allows science to progress.
We're going to talk with other people at this conference today about that issue. So we're going to just skip gears here and go to the next big issue, which is called stem cells. And with our study, we found out that there's actually two types of stem cells: adult stem cells and embryonic stem cells. And can you add your read on what those two things are?
Well, the embryonic stem cells are the most promising kind for future therapies, and also for understanding how cells become different in an embryo, because those very young stem cells have all the potentialities of developing into anything still available to them. Adult stem cells are limited in their repertoire to what they can produce. Both kinds of stem cells are important. Both are, however, needing a great deal of research before they're going to be useful.
Let me quickly, I hate to interrupt, but I do have to ask about adult stem cells. Do you have adult stem cells in your body right now?
Yes, you do too. We all have adult stem cells.
All right, I want to give my adult stem cells to somebody. How do I do that?
Well, you could give your adult stem cells to somebody. Somebody would have to take out whatever tissue it is, say your blood, pass it through a fractionation procedure to get rid of the cells that are not stem cells, and take the small fraction of cells that are stem cells and then put them into another person. The problem is, when you do that, unless that person is your identical twin, you're going to likely have immune rejection problems. So the promise of other kinds of stem cell research in the future is to try to either modify those cells in some way and create a permanent source of material that can go into a wide range of people, or you know, the more far out scheme is to get to the point where we could actually take an individual patient's cells and create their own stem cells, which would be genetically identical to them. That's at least 10 years away. So
okay, well, it's all supposition at this point. But well, let me ask this then. Then what's the value of embryonic stem cells above adult stem cells?
Well, the value of embryonic stem cells, they have much more potentiality. They could create a lot more different things.
Why is that?
Because they haven't already gone down the path that we call differentiation.
Yet. Right.
And once you go down the path of differentiation, many things are shut off to you. It's like me. I'm an adult. There's certain things I can't do. I can't play football anymore. When I was younger, I could. So the same kind of thing about adult stem cells. They have certain abilities and they lack others.
Incredible. Okay, well, we're going to talk to other people about those issues too. But the next one that we're really interested in is DNA vaccines. What's a DNA vaccine?
Well, a vaccine is something you inject into people to make them resistant to another infection, to an organism, either a virus or a bacterium or a yeast cell. Most vaccines are made out of proteins because that is an actual target. The DNA vaccine backs that up one step and injects DNA into people. The DNA then has to make the protein that then creates the vaccine. So it's a longer process. It's a process that works reasonably well in mice, but it doesn't seem to work that well yet in humans. And we're trying to develop it so that it would work, because it's a lot cheaper and easier. You don't have to. DNA is a much more stable entity than a protein. So, first of all, you're talking about vaccinating a large number of people in Africa, etc. DNA vaccine could be a major breakthrough for human health.
Let's talk about something current. We have a big ta-da going on right now about something I had years ago, which was called the smallpox shot.
Right, yes.
And now there's some concern about that. Was I just lucky that I made it alive through my smallpox shot when I was 12?
Well, we weren't lucky because the incidence of harm is low. I mean, we were willing to take that risk. It depends on whether you think the risk is worth the benefit. And I gather today, because of the fact that this is a live vaccine, many people think the risk is not worth the benefit because they're not convinced they're going to have to be protected.
One of the things that's real interesting, you just hit on something. I didn't want to go here, but I've got to ask you this. Live vaccine versus what? Dead vaccine?
Well, or protein, just protein.
Because in today's world we live in, when you get a tetanus shot, you get a live vaccine. Where when I was growing up, you got, I guess, the old dead kind.
Well, I don't know the tetanus story, but live viruses, when they're used as a vaccine, are often much more powerful than a protein because they replicate for a while and multiply and give you more reaction. But in the case of the smallpox vaccination, it's actually infectious to your young children. I have a son who's an emergency room doc. He's got a 2-year-old son. He's supposed to get his smallpox vaccine, which means he shouldn't hug his son for a week or two, and he's not sure his son would understand that, so he's waiting.
Interesting. Okay, we're going to talk with other people about that issue too. So now we're down to the last one, and that's bioengineering. And bioengineering takes in so many different things. For example, changing the way people are, changing their hair color, and changing their eye color, and a variety of other things. But bioengineering, tell us about bioengineering. What is that?
Well, bioengineering is really a broad team. It covers everything from artificial hearts to artificial kidneys to artificial skin. That's one kind of biological engineering, and I don't think that's controversial. The more genetically based bioengineering would replace your cells with new cells and reconstitute a liver from cells from scratch, rather than having a plastic liver, an artificial liver. And we're not there yet. We don't have the technology, and the real debate now is whether scientists are going to be allowed to pursue the technology, because a lot of it involves embryonic stem cells. A lot of the promise involves working with embryonic stem cells. But I want to emphasize that, we're talking about progress that could be made 10 years from now or 15 years from now. I think it's misleading to tell the public that, if we go ahead with this research now, you're going to have something tomorrow. And scientists sometimes get misinterpreted, and I think we shouldn't overpromise. On the other hand, of course, you don't allow this research, it's going to put it off by 25, 40, 50 years.
Let me follow up because you've expressed your concerns about education. And my first question is science education at a crucial turning point, in your opinion?
I think we have a great opportunity in science education because we have enough examples of how to make it work for children that we know how to do it. The problem is, in order to do it, you need extensive investment in teacher training, because teachers never were learning and never were taught science this way, and now we're asking to teach kids how to do science by investigation, inquiry, and so there's a great opportunity. The threat is that the No Child Left Behind Act has a requirement for testing every year in math and English. Right now, that is the sole focus of school districts. And my school district that I worked with for so many years in San Francisco, basically the teachers who taught science, many of them not teaching science anymore because what's being measured is math and reading. So these are elementary school teachers, and they're focusing on what they're being held accountable for. So now, in 2007, the new No Child Left Behind Act requires that there be a test in science. I think that's a good idea, because if you're not testing, you're testing everything else, who's going to teach it? So the really critical thing is, can we develop the kind of science tests that will drive good teaching, or are we going to end up with a cheap, inexpensive science test, which is all the science words, which will ruin the kind of science we want to talk about.
Let me come back to that point. How do we compare with other nations in science education?
The international comparisons, the third international math and science study showed that in 4th grade, our kids are at the top. In 8th grade, they're in the middle, and by the time they graduate from high school, they're at the very bottom. So the answer is the net product at the end of schooling is not competitive.
I have one last question for you there, Larry. I got to ask this. I'm going to revisit that last bioengineering question, and you made some very cogent points there. But the one I want to know is, why is it necessary to collect people's DNA? For example, police departments and the military and a variety, let's say insurance companies. Why would it be necessary to collect people's DNA at this point in time?
Well, DNA collection is used for many different purposes. Right now in the United States, you can only take a person's DNA if they're a suspect. You have reason to suspect that they've done something wrong. And we are building up a large file of DNA sequence information of people who've committed crimes or been thought to commit crimes. And if you have that database and somebody does something, rapes are very easy to detect this way. You could certainly find out who did it. Some people, like Jim Watson, have argued that we should have a national database, that every child who's born, I think Francis Crick would say the same thing, should have their DNA sample taken. And so then, in that case, whenever there was a crime that you could collect some skin or human sample left at the scene, you'd be able to find out who did it. And part of the argument is that that would also tend to inhibit people from doing things because they would know they'd be found out. On the other side of the argument is the general public is worried that if the government agencies have this information, they might use it in other ways. And so this is a big debate that's going on in this country, and I think, 50 years from now, we'll be in a very different position on that matter than we are today. It's going to take time to resolve that.
Let me follow up on that. And the question is, how does the human genome impact employment and insurance issues, and how can we protect people's private information? I think that's what you're saying.
Yes, well, the real problem in the United States is that we don't have universal health coverage. If we were in a place that had universal health coverage, then most of the problem would go away. And so the problem is that if you know your genetic tendencies from your DNA sequence and don't tell your insurance agency, then you could game them by taking in lots of insurance and being pretty sure you're going to get sick and have a lot of money coming in, whether it's life insurance or health insurance. That is really inhibiting the calculations that the insurance companies make when they charge you a premium. On the other side of the question, if the insurance agencies have that information, they could refuse to insure you. So this is a big dilemma, which we wouldn't have if, in fact, we were like in the United Kingdom, where everybody had health coverage. And I think there's a general feeling that we're somehow going to have to get the universal health coverage.
Well, let's ask the final question here. National Academy of Sciences in Washington, D.C. Are you getting enough funding to do what you need to do, sir?
Nobody ever gets enough funding to do what they need to do. We do not get any government funding in appropriations. We get asked one question at a time. We're a non-governmental agency, and each study is a separate contract with the government agency to pay for that study. And, I think we're doing a lot of work. We could always do more. The big issue is not money to us. The real issue is getting asked the right questions. We'd like to get asked, and we are getting asked, harder and more central questions. The government comes to us partly because Congress doesn't trust the administration, so they put it in the legislation to make sure that the right information is provided to the administration. And on the other side, the administration is often trying to protect themselves from Congress. So we're very lucky in this country to have that tension, which asks both sides to ask the Academy what is the truth based on science and technology.
Dr. Bruce Alfords, thank you so much for joining us on The Cutting Edge. A real pleasure, sir.
Well, thank you.
Well, Dr. Kenyon, welcome to The Cutting Edge. What brought you to this 50th anniversary of the Double Helix conference today in San Diego?
It was our work that we've been doing on aging.
You've been working on aging?
To identify genes that control aging. Yes, and we have.
And, everybody wants to find that magic bullet, the Methuselah gene or whatever it is. Does that exist?
Well, we can change single genes in the little animal that we work on, which is a tiny little worm, a little nematode that lives in the soil. You can barely see it with your eye. And it normally lives for only about 2 weeks. But if we change one gene, then it lives twice as long. And it's not just that it checks into the nursing home and just hangs on. It actually stays young for much longer time.
I guess everybody would like to know that because so many people will tell you, they're thinking about DNA therapy and what can happen and how long you can live much longer, but they all say, I don't want to be 160 years old if I look 160.
That's right. This is like a change that, it's as though you look at me and I look like I'm in my 40s, but I'm actually 90. That's what it's like. The worm looks like it's a young worm, but it's really, in terms of the number of days it's been alive, it's quite old.
How do you apply that? How did you how did you create or inject that
Well, we just changed these genes in the laboratory, but it turns out that these genes, they don't just control the lifespan of this little worm. They also control the lifespan of larger animals, like fruit flies, which are much bigger, and mice, which now you're getting kind of close to humans. These are mammals like we are. So, and even actually single-celled organisms like yeast are controlled. They have a lifespan, and it's controlled by the same genes. So this is it's a very ancient system, it turns out, that regulates the lifespan in many animals. And so what's interesting is what the genes actually do. They actually control hormones. So it turns out that all these animals have hormones in them that speed up the aging process and make them get old quicker than they would otherwise.
One of the questions everyone will tell you, of course, is with the Viagra craze and with the HGH craze of hormone that everyone's taking now, these designer vitamins we call them. Anyway, tell us about what is that?
In this case, the hormones are bad for them. They actually speed up aging and make them get old faster.
Really?
Yes, they do.
So you would suggest it's not a good idea to take those.
Say be careful.
Be careful because it will speed
The
up.
hormones are different than one another.
Right.
These hormones that we've implicated in the aging process, they are most similar to the human hormones insulin, which regulates food uptake into your cells.
Right.
And another hormone called IGF-1, which regulates growth. And what we've found is that these hormones are also regulating aging. And there are two studies recently done in mice where people changed the genes in the mouse that affect these hormones in a similar way to the way we had changed them in the little worms. And it makes the mice live longer.
Let me ask you about the consequences of increasing the lifespan. Are there any secondary hormonal changes that occur because you've altered the DNA?
It's very interesting. These older animals in the mice and the worms, they actually seem very healthy. They can be completely fertile, have completely normal reproductive timing and number of progeny, and they seem very,
So the anecdotal evidence says that you can increase the lifespan of animals. And what about plants? What about anything?
It's possible to do similar things in plants. Now, I don't know about the genes controlling lifespan in plants, but at least some plants, if you take the reproductive organs off the plant, then they live longer, at least for some plants. It's as though the plants have a program, some plants, not all plants, but some, reproduce and then die. So you can alter that program.
Well, let's talk about quickly, just 1 second, just on the biological clock that every woman talks about and is worried about. Men seem to go through what they call the midlife crisis. Women go through the change of life. How could you alter that? What's in the future for that?
I don't know actually yet about that. We don't really. I don't have special insight into that at the moment. It would be surprising if the kinds of research that people studying aging are doing now, changing genes or things like that, it would be surprising if they didn't ultimately, learn more about these transitions that would allow.
So how long have you increased the lifespan of these little worms that you've been working with?
Worms that live 6 times as long as
times.
6 times as long?
450-year-old person. So we have worms that normally normal lifespan is 20 days. We have worms that are 144 days old that are still moving and just looking young.
Incredible. Thank you so
I've got one question
Yeah.
for you. Do you feel there's undue scrutiny and regulation of your research of the aging process?
I think I should go to give my talk. Actually, not get into
Thank you very
Thank you
much.
so much. Dr. Theodore Friedman from the University of California, San Diego. What brought you to this conference today on the 50th anniversary of cloning, or of DNA find?
Well, it's a momentous occasion in science. What happened 50 years ago, and there's an opportunity that comes along very rarely to celebrate that. So I'm part of the genetics community here.
One of the things that you're most interested in is gene therapy. Could you tell people what gene therapy really is? What is that?
putting it in right now.
Gene therapy is a potentially important new area of medicine, which diseases are treated at a therapeutic level, not with drugs and medicines, but with genes themselves. We think that most human diseases are caused partially by defects in genes, some of them entirely by defective genes, others partly. And if we can figure out how to fix those defective genes, not to develop a drug to bypass the effect of the gene, but to fix the genes, then we've got a leg up on the definitive treatment of disease. That idea emerged maybe 30 years ago or so. It's been very difficult to translate it into effective treatment. It's been a very complicated area. But I think now in the last couple of years we've seen what is almost surely the first real success.
What were the things that made this possible? What were the benchmarks that made these
Clearly, one of the most important things was the discovery of the coding molecule for all of our traits, that is DNA. How it works, how it replicates. A series of events that came after that involved learning how to move a gene from one cell to another, to introduce genes into cells.
How do they do that?
Well, you can move genes in a number of ways. One way is to insert a gene into a virus. Viruses their job description is to put genes into cells. And if we can remove the genes that they carry that make disease and replace those with genes that may fix disease, then you have what's called a vector, a Trojan horse that puts potentially therapeutic genes into cells to fix a genetic defect.
Now, when you're talking about that therapy, are you talking about years ago? There were wives' tales about women, about being able to grow legs and grow arms on people and supply missing areas of people's bodies and brains and so on. Is that part of gene therapy?
No. That's far from what we have in mind. What we envision is very specifically identifying the genetic defects of a disease and then fixing that in the appropriate cell.
Could you explain that for just one thing? Let's say a child is born with a hole in its heart, for example.
That's more complicated than the initial approaches. I can tell you the example in which it seems to have worked, and that's children who have a gene defect, a defect in a gene that regulates how the immune system functions. Those children have bubble boy syndrome, so they have the susceptibility to disease, to infection, life-threatening. It's been now discovered how one can put the normal copy of that gene into a virus. Take that engineered virus, which is disabled otherwise, but now carries the therapeutic gene. Use that virus to infect cells from patients with the disease. That new gene gets expressed in these children, and they are immunologically correct.
How much of this gene therapy is, I guess, considered DNA vaccine? Is that would you?
DNA vaccine lies in the area very close to gene therapy. It's disease prevention, using bits of DNA that replace a whole organism, a whole virus or a whole bacteria that's previously been used as the agent for producing an immune response. Now we're taking only bits and pieces of the genes responsible for the pathogenicity of this agent, and using that to produce an immune response.
Let me ask you one question, an area that we don't hear too much of lately, and that's Parkinson's. Are we close to coming up?
If you don't hear much about it, you're not listening.
I don't mean it that way, but we're looking at AIDS, solutions for the AIDS problem and cancer. You read about that, I think, more so. Cystic fibrosis we read about.
Well, there's a lot of interest in Parkinson's disease and brain diseases in general. Many of them are related to genetic defects, have genetic defects, and people are working very hard to try to understand how to correct those genetic defects in Parkinson's, in Alzheimer's, in other degenerative CNS diseases. There's a lot of work going on in that area. It's very, very difficult, but we're optimistic that too will work. But Parkinson's is certainly one of the favorite.
How close is that to having something that's been tested and proven out? Where
He's asking if there are cures for some of these things.
No.
There are no cures yet.
Not yet. At the genetic level?
Yes.
No. But all of us are betting that they'll come. They come in your lifetime or my lifetime, I don't know, but they'll come.
What's necessary? Is it more funding for research? Is it more
Better
interest?
ideas and more rigor in some of the research that's being done.
Could the pharmaceutical companies, could they help focus that energy?
They are helping. They're pushing the field in many ways. They're a very important partner in this endeavor. Academia on one side, pharmaceutical and biotech companies, governmental agencies.
Is it easy for you to communicate with the world now with the internet and all? Are you doing collaboration with Europeans or with Asians or anyone else in the world?
Stays quiet very, very short period of time. We all know essentially what we're all doing. Standard literature, word of mouth, the internet, collaborations all over the place.
So if you were to have a success with a child with bubble boy disease, did you say that was one of the successes?
I think that's one of the successes. It's come at a very high cost because some of these children have developed very serious adverse effects. In fact, 2 of them have developed leukemia. So that's not good. That just means we still are very, very imperfect about what we're doing.
Well, so there's lots of road ahead for us to go down.
No dearth of ideas and problems and obstacles. Lots of hurdles to overcome.
Dr. Ted Friedman, thanks so much for joining us on The Cutting Edge. Dr. Christine Guthrie, welcome to The Cutting Edge. Tell us what brought you to the 50th anniversary of the DNA double helix here in San Diego today.
Well, it's a good question. I'm not sure why I was invited, since the major program so far has been a wonderful trip back through history of a lot of the original participants in the development of the central dogma, and it led from the discovery of DNA 50 years ago to how it's transcribed and translated. The step in gene expression that I study actually isn't pictured in the central dogma.
What's a step in gene?
It's a step in gene expression. So gene expression refers to, as Watson and Crick of course showed, that all of the genetic information is encoded in the DNA.
Right.
But only certain portions of the DNA are read out and therefore expressed at any particular point in time. And so how the information that's kind of preserved in the DNA gets selectively expressed is how you get to use only part of that information under appropriate circumstances is what's generally referred to as gene expression. So first,
Okay, let's help the poor people out there that have just fainted from all of your
jargon.
Yeah, it's pretty scary stuff when you think about it. There's something called RNA,
Yes,
and
what
that's
I studied.
ribonucleic acid, as I recall from high school. And you have what's called an M, and from what our research tells us, that sends the message of what the DNA is supposed to do. Is that correct?
Exactly, it's that. So it's a copy of a particular portion of DNA, and that information that's now in the RNA, the information that's specified in the linear sequence of nucleotides in the DNA, is by the act of transcription changed into RNA.
So what you're telling us is if somebody's an American Indian, that's how they get brown skin, or how all these kind of things happen because the RNA is telling the skin to be a certain color, or you could be a certain height.
So the RNA is the part that corresponds to what functional products will be made.
Right. So what happens if you've got, everyone talks about DNA as being, well, you've got a bad gene, or you've got a bad, it's like having a bad transmission in the car there, so you have to kind of fix that. What role does the RNA play in this process?
Okay, I mean, there's a long answer to that because mutations do a lot of different things.
Are we constantly mutating?
Sorry?
Are we constantly mutating?
Well, that as well. So, but the kinds of mutations that I think are of most interest lately are those that are inherited. So mutations that you inherit from one of your parents, that results in the defective functioning of a gene, which then can lead to a disease state.
In your study, have you found that that these things are on some sort of time schedule or clock, that when you grow up and you get old, you get wrinkles, or is that RNA telling the DNA to do certain things at certain times?
The RNA is largely considered not to tell the DNA, okay? So in fact, what's called the central dogma, which is what Francis Crick, who this meeting is celebrating, and others here today, were so instrumental in formulating, is that the flow of information is really unidirectional. So it goes from the DNA via the messenger RNA to the translated protein.
Which is the body, right, or whatever portion
Structural
is.
proteins, enzymes, etc.
Wow.
that are responsible for.
So if you've got a bad messenger, you know what they always say: don't kill the messenger. But if you've got a bad, is there such thing as a bad RNA messenger?
Yes. Well, so I mean, the messenger RNA is passive in the sense that if you have a mutation in the DNA, that will appear in the messenger RNA and therefore in the protein. And so
For all those kids out there, that understand lots of computer jargon, you'd say that was sort of the RNA is sort of a mirrored site to the DNA.
Yes. It's sort of, yes.
And what kind of study are you doing?
Well, so the step in messenger RNA that we study is an amazing step in gene expression that was not anticipated in the early days of the breaking of the genetic code, and that is that when the messenger RNA is actually first transcribed, that is first copied from the DNA, it contains bunches of insertions of nucleotides, okay, that will never encode amino acids by the time the protein is made. So there are stretches that interrupt the functional sequences that have to themselves be cut out physically, removed from the messenger RNA, and then remaining bits stitched back in order to give you a functional messenger RNA that encodes the synthesis of proteins.
Well, it sounds like a scary process. Does your body do all this automatically?
Well, so that's what I've been trying to study for many years, exactly. It's when the existence of these so-called intervening sequences was first discovered in the late 70s, it was frightening because it seemed crazy, and it seemed crazy because it was impossible to imagine a mechanism that could deal with that problem. And so that's what I and my colleagues have been trying to understand is how do you have a mechanism that can do that accurately? And conversely, you can imagine that if there are mistakes in the mechanism that's responsible for doing that, that can introduce even more
Problems.
mistakes.
Yeah.
So that's been our challenge.
What's your goal? What would you like to see happen if you had all of the information you needed and all the funding you needed, or whatever it was?
Well, I think the simplest way to say that to your listeners is that it's now known that a very significant fraction of all inherited diseases are actually caused by errors in messenger RNA splicing. I'm particularly interested in studying how that mechanism of splicing is made to be as accurate as it can be. And so my fondest dream, of course, would be able to understand
Why it works.
And use that to fight diseases where it's not working.
So there are malfunctions of this RNA system.
Oh yes.
And we're seeking causes to why that is, whether it's hereditary or whether it's disease driven, right?
Well, why, but also, of course, how to fix it. And
Correct.
To know enough to know how to fix it, you really have to understand how the normal process works. And we're doing that by studying the process in yeast, actually, not humans, but there is so much similarity in the machinery between yeast and you that actually we can use it as a very effective experimental.
I have a question for you that you may or may not have broached, but when people are born, they're in a natural state. As you grow up, you get vaccines, you get a variety of environmental factors, you get a whole lot of things happening to you. You broke your arm or whatever it is. And through each one of those, you go through a mental breakdown, whatever the issue could be. And as you go along through this process of all these events that happen to you in your life, have you observed that there's changes to NRA or RNA or to
DNA.
And to DNA?
Yeah, I mean, the full extent of that is, I'm sure, not appreciated, although now there are tools to better study that. I mean, another speaker at the symposium, Cynthia Kenyon, if you interview her, she actually studies the process of aging.
Correct. But we wanted to know from your point of view what you see in the next 5 years in your regimen. What are you looking at to do?
Well, the thing that we're most excited about right now, that was the subject of my talk this morning, is the fact that we see a lot of changes in the mRNA splicing machinery in just a simple single yeast cell when it's subjected to what for it is stress. So for example, if you grow a yeast cell in the absence of amino acids which are necessary to make protein, or if you grow a yeast cell at high temperature, or if you grow a yeast cell where there's toxic amounts of salt, all of those conditions we have found have significant impacts on the profile of splice genes that you get out. So we're very interested right now to understand how in this simple model organism does the splicing machinery respond to stress, which I think is very related to your question.
Very interesting. Well, listen, we want to thank you so much for joining us on the Cutting Edge today, and they're very lucky to have you come in and speak to them at this conference.
Oh, you're very sweet. Thank you very much.
Thanks.
Dr. Paul Berg, visiting the University of California, San Diego today from Palo Alto, from Stanford University. Is that correct?
Correct.
So tell us, Dr. Berg, you're one of the foremost researchers and knowledgeable people about DNA probably in the world. So could you tell us why you came today?
Well, I think this occasion is to honor one of the giants of our field, molecular biology, Francis Crick. I mean, Francis Crick's discovery changed the world forever for biological scientists. By solving the structure of DNA, it just paved the way for a whole rash of different kinds of discoveries. For example, the Human Genome Project, which is determining the entire sequence of 3 billion of these base pairs, would not have been possible without knowing that DNA is a double helix and consists of the kind of structure he saw.
As they say in the trade, you're in very rarefied air. For the poor taxpayer that's down here that wants to know where he should put his hard tax money, give us your best, because people are getting a lot of different information. They're getting about cloning and DNA vaccines and bioengineering and stem cell research. Tell us what are the benefits? Where are we get this genome broken down? Tell us just the basic. What are the kind of solutions you're going to come up with?
Until now, we've understood disease by looking at the symptoms of disease, and we try to design therapies that are aimed at alleviating the symptoms. What we now know is that all disease is molecular in nature and has a genetic origin. And if it's molecular and genetic in origin, we know where to look, and that's in the DNA. So the DNA is the root cause, or at least the root cause of disease. We can begin to interpret at a new level by looking at the DNA of each gene. So when you have certain diseases, is because there are mutations within those genes that cause the proteins that those genes make to function abnormally.
Well, let me ask you this now. Everybody's going to ask this question. They're going to say, okay, Dr. Berg, we're with you 100% here, but where's my cancer cure? Where's my cure for heart disease? Where's my cure for Alzheimer's? Where's my cure for the, or whatever it is? So tell us how you're going to achieve these goals.
Tomorrow?
No, I didn't say that, did I?
Well, then you're implying that by asking the question the
Well,
way you did.
they're paying their tax
Okay.
money and they want to know.
For sure. And this is a very complicated machine. The human is probably the most complex organism on Earth. And in order to be able to understand it at the most detailed levels, it takes time. And it's dependent on occasional and hopefully more frequent breakthroughs. Breakthroughs in technology that allow you to ask questions that you couldn't ever ask before, or design experiments that you could never do before. For example, the recombinant DNA breakthrough made it possible for us to be able to examine gene structures in detail, which was formerly impossible.
Now, for the people that don't know, recombinant DNA is actually a cloning process, isn't it not?
It was a cloning process, cloning in bacteria, and nobody objected to the use of the word cloning then, although they're in a tizzy now about using the word cloning when it comes to cloning stem cells. But cloning is not a dirty word. Cloning just means the analog being a Xerox machine. You put in one sheet and you can crank out 1 million copies.
All right, what people are really worried about is the designer babies, and they're also worried about the supermen that we're going to make to go out and fight our wars. Those are the things that are, just the moral issues that are beneath the surface. What do you think of those?
I think they're blown way out of proportion. I think people conjure up all kinds of images. If they want to try to deal with those kinds of images, that's their problem. I think we have to deal with reality. As scientists, we have to deal with what is fact and what is fiction. And we have to design experiments that can distinguish those and settle those kinds of issues.
Well, tell us one success story with the DNA
For
and
example,
vaccine.
Well, one of the diseases which is called chronic myelogenous leukemia is a disease that results from a damage to the chromosome of a specific gene. It actually happens when two chromosomes get mixed up and they break and rejoin abnormally. So you have part of one chromosome joined to not its appropriate partner, but a different chromosome. Okay. Now that break point, at the point at which the two chromosomes become joined, this creates an abnormal gene, and that abnormal gene causes these cells to proliferate enormously. Okay, now we know the gene which is malfunctioning, and so it was possible to design an inhibitor that blocked the action of that gene's protein, and that has now essentially cured the disease of chronic myelogenous leukemia. So there's a perfect example where the knowledge of the DNA structure, the structure of the gene, what the gene does, what the protein that it makes does, and why that's driving the cell into an abnormal division.
Dr. Paul Berg, you're going to start charging me by the hour here, so we're going to just say thank you so much for joining us on the Cutting Edge, and we wish you every great success in what you look forward to in the
My
future.
parting word is, this is a rapidly moving field. It is making extraordinary progress, and I think people have to have faith that out of that progress will come new modes of medicine, new understanding for both prevention, for cures, for therapies, and most of all, from the scientist's point of view, greater understanding.